Well, welcome everybody. I'm emceeing again today. For those of you that don't know me, I'm Darren Johnson. I'm director of the Material Science Institute and a faculty member in the Department of Chemistry and Biochemistry. So welcome to our Discovery and Impact, our monthly Discovery and Impact Symposium series between Thermo Fisher Scientific and U of O. It's great to see everybody again. And we were just talking ahead of time that keep your eyes posted, but we might be allowed to do this in person again soon, and probably can still do a Zoom option for those of you that are not in Eugene or don't want to join or can't join in person. But hopefully this fall, we'll start that off. So it's my pleasure to kick off the program today, introducing Annie Zemper, who you can see probably on the top row of the video screen. She's top middle of mine. Annie attended college in Portland, Oregon, and stayed in Portland for her PhD in cell and developmental biology, which she received from Oregon Health and Science University. Then completed postdoctoral fellowship in gastroenterology at Vanderbilt University Medical Center, and started up the Zemper lab at U of O in 2015, where she's an assistant professor of biology and also a member of the Institute of Molecular Biology. I noticed one thing in your bio sketch, Annie. I really appreciate it. You have a strong focus on the trainees in your lab and what they do and what they've gone on to do, which I love to read. And some of you may have read about that. And there's a diversity of roles those lab members have already started playing, even already in government policy to pharmacy, and many are going on to graduate school in biology and also in medicine. Annie's also an associate member of the Knight campus. And on a personal note, I miss getting to see you at those meetings. We haven't been doing those in person as much. And also we used to run into each other at women's Ducks basketball games and our kids' soccer events. So hopefully the pandemic will allow us to have those chance meetings again as well. Annie's talk today will be Tales from the Crypts. What lessons can we learn from mouse models of inflammatory bowel disease? So Annie, the screen is yours. I think screen sharing is enabled already.
Great, thanks Darren, and thank you to this partnership for allowing me to talk a little bit about our lab's work today and Tales from the Crypt, if you will, telling you how we might learn something from mouse crypts that helps us understand how human disease may work. And I'm waving here to some of you that are my U of O colleagues who I have not seen in a long time. I saw Laura briefly when she came on, and now she's got her her video off, but hi. But it's very exciting to see people, and even maybe people who are here on campus. So okay, so I'll go ahead and get started here. And hopefully this is less awkward than when I try and teach. Sure. Okay. And minimize. Is that, we're good? Yeah. Okay. Trying to minimize that. Hold on.
Looks good on my end, Annie.
Yeah, I'm trying to move my window so that it's not covered, like the gallery of people is covering half my slide. So stand by. And if you've talked to any student who's taken a class from me, they know that I am technologically challenged, despite being what apparently is being the very first year of millennials is what I am. So I don't know if that's a good thing or a bad thing, but I think I'm supposed to know more about technology than I do. Okay, well, it's gonna be blocked, and that's just gonna what we'll go with. Okay, so thanks again for letting me come and talk to you today. So I always start my talks or try and start my talks with the acknowledgements. So you're going to hear about a lot of work today, one tiny piece of my own work and a whole bunch of my trainees' work. This goes from everybody from my lab managers, Bree and Nick, to my 3 graduate students who are getting their PhDs in biology and are at different stages, as well as a fantastic undergraduate who emailed me as a freshman and didn't know a lick about this type of science and has now joined us for a summer fellowship as a sophomore, stayed with us doing lab meetings over Zoom during COVID from his dorm, and he's really been a big contributor, and so I'll tell you a little bit about the work that he's been doing. Okay, so my lab, if you're familiar at all with the things we've been doing over the past 5 or 6 years, you know that we focus on gut regeneration. So of course, this is a cartoon here on the left of a human intestine, and a lot of tons of studies, and rightfully so, are focused on the small intestine, and often a lot of mouse model studies are focused on the small intestine. Our lab tries to focus entirely on the colon. So the middle panel there allows, I'm gonna see if I can get a I've lost my cursor, and so it's hard for me to point to anything. Apologies. There's a schematic here in the middle that is showing you, of course, what we've seen our colon drawn like. But what you may or may not know is that every single your colon is full of these U-shaped invaginations called crypts, and those are depicted there. And they're sort of like a tube. And what happens is that over time, in you and in me and the mice that we study, we regenerate our colon basically once a week for you and I. So if you can think about that, you're completely turning over your full skin lining of your gut once a week. And you can think about that just the way that you slough skin cells. It's all the same continuous tube, if you think about it. And that's the way it happens in development as well. And so we're sloughing that once a week. And I read something when I was interviewing for jobs a million years ago that said that if you live to be in your 90s, you will slough 6 tons of epithelium down the chute, as it were. And so that's an incredible, if you're for a cell biologist, I think that's an incredible amount of proliferation and cell movement and cell turnover. And if you think about it, it's surprising that we all don't have some sort of cancer by the time we're with our gut, by the time we're in our 20s, okay? You can live to be in your 90s and you're sending 6 tons of epithelium. And so this is just an incredible system. We focused on both normal states as well as in disease states. In particular, we focus on preclinical models of ulcerative colitis or generally inflammatory bowel diseases. So we all probably know that chronic inflammation in our gut is uncomfortable, but it can really lead to long-term disease. So all of those epithelial cells that make up those U-shaped invaginations I just showed you have these connections between them. And that acts as a barrier between the luminal contents and the rest of our body. And so when that barrier is disrupted, what we end up with is our inflammatory cells coming in, sending signals to ameliorate that signal that's there, but it also ends up with a lot of cell death, okay? Both in the epithelial layer itself, okay? So our inflammation in our gut actually can lead to this phenotype that I'm talking about. It can really lead to reduced peristalsis. So our gut slows down, we don't feel well, okay? That means that the intestinal contents aren't moving as much, which can then lead to infection, which it can then lead to more inflammation, and around and around and around the circle we go. And it turns out that colon cancer is the second leading killer of males and females together in the United States, behind cardiac-based diseases. And if you have gut inflammation on any chronic level, you are 6 times more likely to develop colon cancer, okay? So this normal regenerative process of being able to keep our gut healthy and a tight barrier is really important. We know that 1.6 million Americans have inflammatory bowel diseases, and there are basically 70-ish thousand new cases of inflammatory bowel disease diagnosed every single year. And this is an old graphic down here on the right, but it's from the CCFA, the Crohn's and Colitis Foundation website, that tells us that over time we've been able to, in the advent of being able to sequence genes and such, we've been able to identify much more than, I'm sure, 163 now associated genes with inflammatory bowel disease. And so this is a rich area for us to try and understand what's going on both normally as well as in inflammation, and then maybe try to think about prevention strategies so that we don't get to that point where we have colorectal cancer. So I made the point about cell proliferation within the gut. The stem cells are at the bottom of these U-shaped crypts, and they're labeled in brown there. They are the workhorses of this system. They churn out daughter cells, as we call them. And this graphic on the left is a cartoon, but on the right, this is a beautiful image from Duke that's used immunofluorescence to just label different proteins within the colon and be able to show us sort of the diversity of cellular phenotypes that are there. Of course, this is a great system to study different aspects of cell biology and regeneration, such as proliferation, migration, differentiation, cell-cell communication, senescence. There are senescent cells within the gut, as well as cell death. Okay, so this is why this is the best system, in my opinion. So we use genetic, chemical, and molecular preclinical models, mouse models, to examine gut regeneration and disease. And so that's my little schematic there on the right, or on the left. We are able to do standard things like take out the gut and put antibodies on it to do immunofluorescence. We can grow it. This is a collaboration here that we started a number of years ago with Raghu Parthasarathi that allowed us to use his light sheet microscope to look at these cells over time. We can, of course, extract the crypts and look at them in 3D. And then lately we've been doing single cell sequencing within our system. So this is just a flavor of the types of approaches that we take in my lab. So how in the world can we use these preclinical models to give us any clue about human disease? Okay, so on the top left, and I apologize for not having a cursor. On the top left, we have what's called an H&E, and this is a hematoxylin and eosin stain, and you can see those little U-shaped invaginations, and those are crypts. When we have inflammation, as I mentioned, the epithelial layer oftentimes will completely apoptose or undergo cell death and go away. And so what happens is that you end up with the histology that's just below that, where you can't really see any well-defined crypts anymore. What is amazing about our gut, in you and in me and in the mice study, is that over a very short period of time, the gut will regenerate itself to look like the top panel again. Okay, and this is, you can imagine this is a highly dynamic process. You can imagine that there are many different stages to it, and all of this we don't know a ton about. Okay, but these are the things that we're trying to learn. So of course we can take these tissues and we can stain them for our favorite markers. That's shown on the top right, where normally that's labeled as control. We have this protein of interest that's just in some of the cells at the bottom in green, and we can do different treatments and actually look for an expansion of that marker in these different perturbed states. And that gives us information about what types of things molecularly are going on that are different from in homeostasis. This is a graphic here that recently came out in one of our favorite journals, Gastroenterology, that really just I think harnesses the power that we have in our system, is that we can take normal colon, we can cause inflammation, and then dynamically over time look at regenerative processes, look at things that speed up regeneration, look at things that impede regeneration. And so generally in any type of preclinical study, what we want to do is map the normal process, map the perturbed process, so in this case, inflammatory states, determine what is required by that process, about the perturbed, or I can't see my questions, but determine what is required for that process, and then determine how these things are changed in the perturbation. And so I've been doing, I joke that I've been staring at mouse intestinal crypts since 2004, which isn't a joke, it's actually for real. But I've been looking at the colonic crypts now since 2009, and when I was doing postdoc. And I've been spending a lot of time when I was a postdoc, and now my lab spends a lot of time trying to identify key molecular players in gut regeneration and defining their roles in normal gut regeneration. So you don't need to know anything special about the intricacies of the data that's on the left, but suffice it to say, we spent a long time identifying a stem cell and progenitor cell marker that we can now track in the gut that helps us understand those specialized cells that are really the workhorses of that regeneration. In my lab now, we've gone on to perturb the mice, give them this inflammatory state, and ask, how does that progenitor cell population behave differently? So we can give the mice this inflammatory assault. We can look 1 day, 2 days, 3 days, 4 days later. We could even look 3 months later if we wanted, to understand both the short and long-term implications of colitis on this really important progenitor cell marker. We've gone one step further and done single cell sequencing of our normal gut and our inflamed gut, and we found some really fascinating differences. So again, we don't need to understand all the intricacies of the slide that's on the left, but suffice it to say, this is the cellular signature, or what each cell is doing at this given time when we compare our normal colons to our inflammatory colons. And unfortunately, my circles moved a bit, but there are 3 key populations that we focus in on that show up in inflammatory states and are not there normally in homeostasis or in normal conditions. And so we're really focused on what these new populations are. Are they representative of some sort of transition state? Oh, here we go. Here are my questions. It's almost like I planned this in the talk. What are the newly defined cellular populations? Are these unique to inflammatory assaults or other types of, for instance, an infection or a pathogen? Maybe these show up then too. Do these represent a general cellular phenotype for any regenerative tissue that's in stress? So would I find it in my skin? Would I find it in my blood? Cells that are like this? And how does this compare to human biology? And of course, our clinical partners are always coming out with these amazing resources that let us compare our mouse studies using really similar approaches that we take in the lab and do them for patients. And then we have these huge data sets that we can compare our data to. And so we're always doing this in the lab as well, looking at, so this particular study is actually looking at normal, adjacent normal, and early inflamed gut and saying, what are the cellular changes? This is the exact same thing we're doing here in our lab. And it's really nice to be able to see what the overlap is between the preclinical model and the clinical study itself to help us hone in a little bit further to understand what might be important for human disease beyond the simple, beyond the very complex, just normal biology that we're observing. So we also take approaches where, for example, we can use preclinical knockout mouse models. So this is mice that are ablated for a certain gene of interest, and then add in inflammatory assaults and help define the molecular requirements for regeneration, both in normal times and in disease. So this is one observation that my graduate student, who's going to be a 6th year this year, made when she first came to the lab, that we have these weird mice. Their colons are extremely long. They're very long, and it had not been reported before, but okay, that's an interesting observation. What's actually happening there, and does that set the mouse up for some other issues later on, and what can we understand about sort of the molecular role of that gene? So we went on to characterize this, and she's done a lot of work. Again, don't need to know everything that's in the panel here, but suffice it to say their colons are longer. They have taller U-shaped invaginations. They have a significantly higher number of cells within that, within those U-shaped invaginations, and they have molecular and signaling differences, okay? And so what we found is that this particular knockout, as I mentioned, is sort of setting itself up in development to be different than in homeostasis. And so, well, what happens if we give this mouse inflammation? So again, we can take this mouse that seems to function normally, but has this odd, super long colonic phenotype, and actually give it inflammation. And that's what the schematic here on the right is showing. And then we give it for 7 days. Then we can give the mouse water and let it recover, and then see sort of what type of molecular changes are happening within that mouse compared to the normal mouse. And so what we find actually was really, really shocking. And this is really surprising because our gut in you and in me, and again in the mice that we study, have built-in functionally redundant systems, so that this typically giving a short bout of colitis to a mouse doesn't do much to them other than perturb their gut for the short amount of time that we want to see it. Unfortunately, what we found is that this particular knockout mouse shrinks its gut aggressively, and it cannot recover to the normal state. These mice actually have to be sacrificed before the end of the study because they can't survive the inflammatory assault. So there's something about this particular knockout, this gene, it's called LRIG3, that's required for the survival of these mice from the colitis inflammatory state that we give them. And this is the project that my graduate student and my summer student, Kevin Mueller, have been working on. He's the one that emailed me as a freshman and has come into the lab and is just really whipping through looking at this phenotype. We find out that despite the fact that the colonic crypts were taller, they end up much shorter, despite the fact that they have many more progenitor, these stem cell workhorse cell populations normally. they actually have almost none after you give them inflammation. And so we're really interested in the mechanism of this and to see whether or not this relates in any way to the type of gut failure that a lot of colitis patients see. So hopefully I've been able to give you a flavor today for some of the things that we work on in my lab. We, again, are using these preclinical mouse models. Wasn't able to tell you about our projects in development as well as in homeostasis, looking at cell-cell communication. But those use the same strategies I mentioned, as well as fluorescence activated cell sorting and tissue culture to be able to ask, so how do these models compare to humans? How can we employ genetic engineering approaches to help us prevent gut loss? So you can imagine taking these models that we have and being able to give them add back, for example, a particular gene or ablate a certain suppressor type of gene using CRISPR, for example. What other small molecules or chemicals could we think about supplementing to the mouse to maybe prevent this atrophy that we see in the last mouse, for example, or modulate the normal inflammatory response? And how could we maybe even use these mouse models to help evaluate how our current standard of care protocols for looking at patients and helping patients that have inflammatory bowel disease and how those particular strategies aren't as efficacious as we would like them to be. And certainly this is a focus of a number of labs, both basic science and clinical across the nation, some of which are our collaborators. So with that, that's my short time. So I want to thank, of course, the people that have done the work. So you heard about Bree and Nick and Tim and Janelle's work today. Rachel is working on a project looking at that really important workhorse population in gut development. We have our UO collaborators listed here on the left, our national collaborators listed on below that. Some of them are important clinical collaborators that help us understand what they might be seeing in patients. I mentioned that we have a number of previous lab members. I think I was counting it up and there are like 14 previous lab members from the lab, both from the undergrad level all the way back up. But I've noted the ones here that participated in the work that I showed you today and just mentioning where they are at in their training. As Darren mentioned, a really important thing for me is to be able to send good scientists, citizen scientists out into the world, regardless if they decide to stay at the bench. And of course, our funding sources are here, NIDDK, the American Gastroenterological Association, the VPRI here, and this awesome program that we have for undergraduates called SPUR or OURS, if you are a UO undergrad. And with that, I will stop and I can take questions or we can chat.
Excellent. Thanks, Annie. Folks can feel free to post questions in the chat and we can read them out, or feel free to unmute yourself and ask questions. We're not too large a group, I don't think, for that. And Annie, I see one in the chat already. You want me to read them to you, or do you see the chat? Sometimes it's hard with this.
Amazingly,
Okay.
I see the chat. Once I got rid of my own screen, I actually now have a cursor and I probably a laser pointer. So it says lectins can increase the rate of epithelial cell turnover in the colon. How does that affect recovery from inflammation? So I know for sure that various lectins, as well as different oligosaccharides, completely change, especially if a mouse is sort of immersed in that type of diet, change the recovery rate. So normal cell turnover, and it is also changed within the context of inflammation. I can't recall off the top of my head the specifics. I haven't done any studies like that, but that's definitely something that people are looking at. Of course, dietary changes are part and parcel to what a lot of people do to evaluate this in mouse models, because that's the easiest thing we can change as humans. Any other questions?
Yeah, hi Annie, this is Dave Piper from Thermo Fisher.
Hi.
Nice talk. Thanks for sharing your work with us today. I was wondering, I know you work in the mouse model and it's obviously got a lot of benefits in that you have the intact animal and the intact organ, but how do you view some of the emerging iPSC related organoid cultures as a means to understand human pathophysiology and compare it to these models?
Yeah, it's a really good question. So I think the short answer is they are awesome but limited, but they really are able to take advantage of something that we can't do very easily. So our partners that work in zebrafish or the fly, for example, or have a lot more accessibility to the gut than we do in terms of imaging. When we look at a mouse and we give it a treatment and we want to look at X or Y, and especially in the colon, just because the colon in the mouse actually happens to be up near the back of its spine in the rear, you have a really hard time accessing it to be able to do anything over time, for example, like imaging, and it's really stressful to the mouse, and usually you just have to kill the mouse and then you have a time point, but you don't have anything that you can look at over time. So induced pluripotent stem cells offer, especially when they've undergone directed differentiation into colon, for example, or small intestine, really allow us to understand how cells make decisions with it in a really, really zooming in on it. But in a lot of cases, although this is getting more expanded now with some work out of Michigan, you're missing a lot of cellular signals that might come from cells around it. So within the colon, we have a 3D microenvironment that is full of laminins, for example, and collagen. So we have physical pressure on the epithelial cells. We have blood cells there. We have mesenchymal cells that are, offering cellular signaling. The whole thing is moving, right? We have a whole 2 layers of muscle pushing things. And so I think we can learn a lot from IPS studies, but it's really restricted in terms of, I think you then have to go to the experiment in the mouse and see, how they might complement each other.
Yeah, thank you. And I guess then connected to what you just said about the full environment and some of those interactions, have you been thinking about moving your single cell RNA-seq work to more of a spatial approach to understand what's happening from a cell to cell, spatially located viewpoint?
Yeah, so one of the things that I was involved in right before I left my postdoc at Vanderbilt was doing these multiplex assays where you could sort of look at 152 targets within the context of the tissue. So instead of doing, 2 or 3 stains, you can do 150 or so. Now these, and I believe that Thermo has one too, they have these systems that you can pick up that will allow you, basically you look at a certain stain and then you ablate it and go do another one and another one and another one. And what's cool about that is when we do our single cells, first of all, we're getting a transcriptional readout, okay, which is RNA doesn't always make protein, okay. Second of all, we're losing all of I like to say we're losing our real estate. That's what I tell my lab. We're missing where these cells are in relationship to each other. So if we can do these multiplex assays where we're looking at proteins and we're keeping the tissue intact and be able to say what cells are next to each other and what does that mean about their cell signaling, that really sets us up better. It also helps complement our ex vivo studies where we take the colon out and grow it in culture. Again, we're missing some of those components that we have within the mouse, but it does allow us to then, for example, if you were to do a multiplex study on an organoid culture, it would give us a much finer lens and we'd be able to manipulate that more and then do multiplex and ask how that changes, rather than on the whole mouse where you have physiology that's going to be mucking up the works. Scott says, can you say something about the senescent cells that you see in the colon? There are certain cells that are sort of reserved down in the progenitor U shape in the bottom of that cup there that are activated in certain injury contexts. And so a lot of people have defined them as being more senescent, and they have certain sort of molecular features of senescent cells in general. Of course, the senescence field is huge. And one of the things that I think people have come to realize over the last few years is that actually the number of these reserve or senescent cells increases as the mice age. And so that might represent some sort of mechanism in humans to have sort of a backup system as you age to prevent gut loss or, sort of misregulation within the crypt. But we don't work on senescence directly, but it's definitely something that the field is focused on and that we can easily stain for within the gut. Do you see a connection between DNA mismatch repair and epithelial. Yes. So there's been a couple of studies. One of them has come out of between epithelial turnover, not colorectal cancer. So, interesting. So I know that there's been a number of knockouts of certain mismatch repair genes, and certainly those mice end up getting higher rates of colorectal cancer, especially if you perturb them and spur them on to do it. I cannot remember off the top of my head what the normal rate, how the normal rate compares, like how frequently the cells go up. So usually it takes about 4 to 7 days for them to go from the base of the crypt to the top. And I don't remember in the mismatch repair mice if that rate has changed, but it's a simple experiment. I'm sure people have done it because you can trace the mice using bromodeoxyuridine and actually watch the cells go up the side of the crypt. I'm sure that rate must be perturbed, but I don't know off the top of my head.
Great. Thanks a lot, Annie. And I think people can ask questions privately during through the chat. And also we have 15 minutes at the end that we kind of keep the channel open. Could extend it longer if needed, if people want to have discussions afterwards, so we can have some additional conversations after the talks as well. So thanks again, Annie, for a great talk. All right, we are going to shift gears to Chris Vonnegut, who's just unmuted himself and sharing his video. Chris is a staff scientist in chemistry R&D at Thermo Fisher Scientific. He got his bachelor's degree from Benedictine College, and then after a short time at Boehringer Ingelheim in formulations R&D, he moved to Eugene to pursue a PhD at U of O. And I should know that pretty well because he got his PhD working on a joint project between my lab and Mike Haley's, where he was focused on synthetic chemistry and physical organic chemistry of inherently fluorescent hosts designed to bind anions. And I got to give Chris a ton of credit, because an offshoot to that project was a surprise that he discovered, and it ended up launching a totally new direction in the lab. So he and his undergraduate coworker discovered a new class of fluorescent phosphorus and nitrogen containing heterocycles that were really potent fluorophores and also had really interesting hydrogen bonding that allowed for some oxoanion recognition. And so we've taken that a long way since Chris left the group, but he gets a ton of credit for starting all of that and also being the key inventor on the first patent on those molecules. Chris has been at Thermo Fisher since 2016, and during his time there has worked on a variety of nucleic acid interrogation techniques from both the synthetic and application side. He's also been an original industrial partner in our new Night Campus graduate internship program track in molecular probes and sensors. And in fact, before the talks got started, we were talking again about an immersion lab that we may run this fall with him. So he was an early partner in that program, helped us with the initial immersion labs, and has given us advice over the 2 or 3 years that program has been running. So thanks for that support, Chris, and look forward to working with you in person this year, hopefully, as we work with the students in that. Chris's talk today will be Modern Nucleic Acid Dyes, Development, Exploration, and Application of a Powerful Interrogation Motif. And Chris, screen sharing is yours.
All right. Thank you, Darren. Let's see here. Okay, is that coming through for everyone?
Yep.
Excellent. All right, Darren, thank you for the fantastic introduction. It's really a pleasure to speak to you all today about the work we were doing with nucleic acid dyes, kind of a little bit of the history, some of the development that we've done for these, and then where we've managed to take this huge interrogation motif that we've developed here. I can rehash a little bit of the introduction. Like Darren said, did my graduate work at University of Oregon. As far as the more fun things I do beyond like professional history, really big fan of brewing beer, making, kind of flexing my chemistry muscles in the kitchen, making various extracts and things like that for cocktails. Darren, it is something that you can't let me forget, is I make a fantastic tonic syrup that I need to share some with you. Beyond that, yeah, so a lot of my work at University of Oregon was based on fluorescent scaffolds, things of that nature. So it's a really nice fit here into Thermo Fisher Scientific. I did a brief internship here during the last year of my PhD work. It really was a great fit with the site, with the people, with my skill set. And so pretty much, came back after the internship to University of Oregon to finish up the PhD work, and then was able to come back in here full time. And I've been working here ever since. A lot of my work is novel dye scaffolds, shifting their spectra, their binding properties, what biomolecules they're actually looking for. And a ton of my work keeps looping back to design application of new nucleic acid dyes. So that's going to be the main focus of my presentation here. So basically what I'm going to go over is kind of a brief history of nucleic acid stains, where they started, where they came from, kind of how they came up through the steps to the huge number of them that are available out there today. Kind of take you through our development cycle as well, from the original like Toto and Topro dye families that we first put out there on the market, then the Cyber dye family, and some other like more interesting applications we've done with these, beyond just binding DNA, RNA, and single-strand nucleic acids. Some activity probes are based on this nucleic acid dye technology. Some of the really cool gel stains we've been able to develop from these same scaffolds, and then some really targeted solution-based quantitation of various structural motifs. And from there, would love to kind of pick all of your all's brains at University of Oregon. What look for some potential collaborations, where you might need some help, where we could use some help, and where we could work together. So to start off with, the classic nucleic acid stain, and I think you mentioned it in your presentation, right? Hematoxylin. It's still currently used in H&E stain. The common counter stain for that is eosin. Hematoxylin is a really good colorimetric stain for nuclei. Largely nuclei. It stains ER and a little bit things like that too in the structure. But for the most part, in the stains in tissue, you'll see the nuclei being stained. And this top left picture here, hold on a second, actually, let me see if I can get my pointer up. Yeah, and so you can definitely see, the nuclei being selectively stained here in this tissue sample. This is human small intestinal tissue. It's kind of an interesting compound. It's been used as an ink or as a stain for plant slides. I think the first reported use of it was back in 1750. Some of the old histochemical preparations of this are actually pretty hilarious because it's the actual stain and the actual dye is not the natural compound here, the hematoxylin. The actual stain is the hematein, which is the oxidized component from the hematoxylin, forms this quinoidal type structure that gives it a good conjugation, good coloration. But some of the original preparations, you actually you add in with a certain number of metal salts, certain pH, and then you let it sit for a month due to the native air oxidation. Then it's fine for use for a couple of years, according to those old protocols. But it kind of shows you how these things came about. These were old, textile dyes, old ink dyes that people started using to stain tissue and histochemistry and started seeing what they stained and then kind of moving on from there. It really kind of came into its own in the late 1800s, early 1900s, and people started paying more attention. There was more understanding of the dye molecules and the structures they were looking at under the microscope. And this kind of led up through to about the 50s through the 70s, where you start seeing some more of the fluorescent type compounds. Fluorescence is starting to be more interesting. People started noticing that as a real phenomenon that could be understood and then utilized for staining applications. Probably the earliest and one of the most widely known fluorescent DNA and RNA dyes would be these phenanthrene-based dyes. Your ethidium bromide, propydium iodide, hexidium. Those tend to be named for the length of the alkyl chain off of the nitrogen for this phenanthrene type structure. These are not very impermeant dyes for labeling like live cells and things like that. They do make great dead cell stains. They've been around forever. People are pretty comfortable with their behaviors. And you can see them here in an example. So this exact picture is bovine pulmonary artery epithelial cells. They're stained with a number of different fluorophores. In this case, it's part of a live dead kit. So the green is with our calcine AM stain. It's basically activated by esterases and shows living cells. And you can see the nuclei are really nice, bright red, because those are the cells that the ethidium bromide was able to penetrate and actually stain. So you can kind of utilize that membrane permeability for some applications specifically, and I'll get into that a little more later. Some of the other dyes that people are probably more familiar with were ones that were developed in the 70s. DAPI, the different various flavors of Hirsch stains. These were both developed in the 70s. They give pretty good staining. They have good selectivity. They are pretty much UV excited, blue emitting. You can see up here in Drosophila embryo, the nice nuclear staining from DAPI. And then in this case is a HeLa cell that's been stained with MyoTracker Deep Red. I believe it's been incubated with a cell light Talon GFP probe, and then as well with the nuclei stain with the blue emitting stains. But what do we have going with these different dyes? How can they be improved? What are the drawbacks? Again, hematoxylin is a nice stain. It's still in use, really largely for diagnostic purposes. It doesn't really have much of a fluorescence profile. So that's kind of a huge part of what we do here. So that part of it's kind of out. That being said, it's still being used really frequently today. DAPI and Hirsch have really excellent DNA selectivity. It's only available in UV channel excited channels. They're really not very sensitive, but they're really solid for what they do. Ethidium bromide, propylium iodide, all of those have decent sensitivity. Their excitation emission channels are a little strange compared to a lot of our fluorophores that have like tighter Stokes shifts and things of that nature. They do have pretty poor selectivity between DNA and RNA. So when you started thinking, like, what if sensitivity, selectivity, and usable channels of all these DNA stains could be expanded? But before we even start talking about that, we got to think about like what are the mechanisms these dyes use to bind to DNA, and how can those be leveraged, right? So the typical ways that things bind to DNA, not just dyes, but absolutely everything is intercalators, minor groove binders, major groove binders, and external binders. DAPI and Hertz are both known to be minor groove binders. There are solved crystal structures of them in the minor groove for short DNA oligos. And of course, that helps with their selectivity, right? Because the minor groove is only available for double-stranded DNA helices. Intercalators are an option, and that's what ethidium bromide, propylium iodide do. Their fluorescent enhancements largely due to hydrophobic effects. Once it gets in between the base pairs and then you have excluded water molecules, you no longer have water quenching of the fluorescence, and fluorescence turns on. External and major groove binders are a possibility for macromolecules, but when we're talking about small molecule fluorophores and dyes, those features are often too large to be utilized with a small molecule motif. So, molecular probes first started looking at these, and I would say, late 80s, early 90s with, our fluorescent dye expertise and molecular design, and really started digging in. There's some improvements we did for, like, you look at like ethidium homodimers and stuff like that to really improve the binding affinity, as shown by these best intercalators here. But what really drove the revolution, I would say, in nucleic acid dye technology was this brand new binding motif called thiazole orange. This was first published in the late 80s by Linda Lee. She was a Perkinelmer at the time. And you can see the structure here for the parent thiazole orange. It's an asymmetric cyanine dye. In free water, it is almost completely non-fluorescent. The quantum yield is very, very low. I want to say sub 0.1%. But whenever it binds to nucleic acids, you get a huge turn on. The quantum yield gets much closer to unity, so you have about a 3,000-fold enhancement of free in solution versus when it's binding nucleic acids. It's got really, really nice fluorescence channels, right? It's really close to fluorescein. It has, so you're able to use the 48 nanometer laser to activate it, and you can see with the green emission, things that people are used to, things that the instrumentation was ready for at the time. It did display some poor DNA and RNA selectivity, but it was a great starting point. So using this dye as a kind of a jumping off point, molecular probes did huge structural modification of these asymmetric cyanine dyes. You can modify the heteroatoms, you can modify the conjugation link, some of the groups around it. And we were able to access the full spectrum of nucleic acid dyes from this motif, right? So you have all the way down from blue emitting, kind of similar to your Hertz and DAPI, all the way up to a couple of members that are way out there emitting at 770, right? Being excited by a 750 nanometer laser. So by taking this one, we were able to start really have a good scaffold that you can sink your teeth into and develop a ton of different nucleic acid stains. So the original ones, the Toto and the ToPro dye family, the first ones we started making were these homodimers, and they have really interesting structures, right? So this is a solid crystal structure of the Toto dye homodimer. We call it a homodimer because it's actually two different thiazole orange units linked by a linker. The linker has some cationic groups on it, so it has favorable interactions with the phosphate backbone of the nucleic acid. And it's actually a bis intercalator, so it actually forms a staple. This has a really, really high affinity for DNA. So you're actually able to pre-treat your DNA with this dye. It'll label it so powerfully that you can then electrophoresis and it stays balanced to the DNA. So it's a really interesting scaffold. But when we start looking further down the road and thinking okay, applications in cells, different applications in biotech space, these dyes are not very membrane permeant, so they've got a lot of charge. And maybe we can do some more stuff to the structures. That starts building out the Cyto and the Cyber dye family. There's a huge number of dyes in the catalog. We've made literally hundreds. It's probably over 1,000 in all honesty of all the different varieties of structures we've made for these nucleic acid dyes. And we've done a lot of screening of them, and we have huge numbers of different dyes of different properties. We currently sell sampler kits for all those dyes from blue emission, green emission, orange, and red. A lot of the members show a huge variety of specific staining properties. A lot of the papers you see published are, we screen these dyes. We found this one had this specific property in our specific assay, and then so this was the result and we wanted to use it for. And then our internal screening, what we've been able to do is, go through, look for their use in certain applications. And I'm going to kind of go through like the various, not so much niche, but interesting target applications we've been able to pull off with these dyes by looking at this huge library and seeing what we can actually target these dyes for. A really interesting application is the Cytox Dye Family. So certain members of the Cytox Dye family are really useful for staining the membrane compromised cells. They basically have a really, really high affinity for DNA, but they don't penetrate membranes. Now, if we couple those with one that does, then we can have a really interesting live dead type of assay for cells. So on the right here, you see a bacterial sample that's been treated with a combination of two different dyes out of the Cyto family. So the red ones are a membrane permeable dye, Cyto59, so that then those are representative of both the dead and the live cells, right? So viable cells tend to have an intact membrane. But then if you have the membrane compromised cells, we treat them with Cytox green. Cytox green, it penetrates the membrane and will label DNA inside of all the dead cells as well. So you can basically assay an entire population and get a good number of live and dead from this mix. Now, if we expand this out to some other different assays, we have here a really interesting trace. So basically 3 different groups of cells were labeled with different cell trace reagents. Now, cell trace reagents are reagents that you can use to label a population of cells, and they will actually stay labeled with the cells through several generations, so that that family of cells will stay labeled with a certain color that you can use for barcoding and things along those lines. In this case, incubated natural killer cells with cell trace violet, CD20 positive Ramos cells with cell trace far red, and CD20 negative Jurkat cells with yellow. And then you mix them 1 to 1 to 1, incubate for an hour at 37 degrees C, and then label the entire population with Cytox green. Then you run them through the Attune NXT for a cytometer, and what you can do is actually measure the antibody-dependent cell toxicity that you get from having those different populations in there mixed with natural killer cells. And you can see the CD20 cells show very little killing in the assay, whereas the CD20 positive cells show 32% dead from this Cytox green dye treatment, which matches about what we would hope to see from that kind of staggered assay, especially looking at the antibody-dependent cell killing. We have these dyes available in a few different colors. People have used them a ton for different types of toxicity assays, drug screening, things of that nature. And so, yeah, we have the blue one, the green, and then a far red and a red. And those have been really useful. You see a lot of different applications and publications of those. Kind of along the lines of cell viability, we have this designed assay called the Cyclone Direct assay. So far, this has been developed in 2 different colors, green emitting and red emitting. And what's clever about this assay is you actually have these membrane permeant nucleic acid stains. They will go into every single cell that's in there, right, whether the membrane is compromised or intact. But then you treat the entire cell population with a quencher that is not membrane permeant. So then the membrane impermeant quencher only gets into the membrane compromised cells and quenches the native fluorescence there. And what this allows you to do is either plate reader or imager, you can essentially measure the number of live cells in your population. You see people use this a lot for pharmaceutical treatments, things of that nature, where you can watch and you can get your EC50 curves from treatment of your cell populations and watching the number of dead or live cells you have there. So here's an example of how you can basically multiplex the far red emitting Cyclone Direct red assay with MitoTracker green, show no real signal overlap or anything like that, which is actually really useful for GFP expressing cells and things of that nature. We've also been able to develop this family out for gel stains. So the gold standard for staining gels often tends to be silver stains. You see a lot more with protein than you do with nucleic acid, but it's used for both. But we have developed, Cyber Green, Cyber Gold, and Cyber Safe, or what we call our flagship nucleic acid stains. They have a huge, huge sensitivity on gels. They're pretty much commonly used now. I would say they're not quite the standard. People are still using ethidium and propydium iodide. I will say, also from personal experience, an RNA gel that you run with ethidium bromide, and it's hilarious. You'll see nice 2 sharp bands, exactly like you'd like to see for intact RNA. You run it with Cyber Gold, and you see a lot of smearing that you simply could not see with ethidium bromide. And it's something I remember whenever hearing tales about when these were first released, talking to customers and then them, I won't say complaining, but suddenly being concerned about what they thought were really, really pure isolation preps and realize how much smearing they had because of the huge sensitivity of these gel stains. Cyber green stain is not just used in gels. We have also enabled an entire platform of qPCR. I have certainly had no time to go into that here, but all of the applied bio qPCR instrumentations initially built using the cyber green dye as a platform to measure the increase in double-stranded DNA as, of course, your DNA gets amplified. And then cyber safe is actually a really nice stain we developed. Ethylene bromide is a fairly strong intercalator. It has quite a bit of mutagenetic properties. We did some tests with cyber safe and showed that it's quite a bit safer than ethylene bromide, and demonstrate a lot less mutagenicity, which is pretty nice. And you don't want to be handling intercalators with bare hands or anything like that. So having something that's quite a bit safer than the Ames test was really nice to put on the market. Another application we've seen for these nucleic acid dyes is actually using its binding and its affinity to DNA as a switch. So we have our solvent Caspase 37 green reagent. What this does is it has a peptide that's actually conjugated to the nucleic acid dye. And the DNA affinity is completely deactivated until the peptide is cleaved due to caspase 3 or 7 being upregulated in the cells. And so that way it acts for a really nice protease switch, right? In this case, we're specifically interested in caspase induction. You can see here a few different examples of it. So on the left is U2OS cells where caspase has been induced with etoposide. On the right is HeLa cells that have been induced with staurosporine. And you can see really nice positive controls or positive results for the pharmaceutically induced apoptosis in these cells. This is really, really cool. We've investigated this mechanism for a few different peptide substrates, and it's always nice to be able to use these type of binding mechanisms for something beyond strictly nucleic acid binding. The ability to quantify DNA, nucleic, and RNA, and your other single-strand nucleic acids has also launched its own complete platform of kits. And so all these quantitation kits are called the Quantit. We have them for a number of different biomolecules. I'm going to mainly cover, of course, the nucleic acid ones here. So we have a handful of nucleic acid dyes that have a huge selectivity for double-stranded DNA over single-stranded DNA or RNA. Greater than 10x signal fold enhancement for double-stranded DNA over single-stranded DNA or RNA. One of the members of the kits has over 25x enhancement strictly for double-stranded DNA over the other ones. And the nice thing is that for a lot of your downstream applications, you need really, really tight control over your nucleic acid concentrations in solution, right? qPCR inputs, sequencing inputs, things like that. So what these allow you to do is one, measure really, really low concentrations of nucleic acids in your sample. And two, it's highly specific, right? The gold standard for a long time for nucleic acid quantitation was always UV vis. But the problem is that you're measuring the absorbance at 260. Free nucleotides all measure at 260. A lot of the contaminants from nucleic acid isolation measure absorb at 260 as well. So this lets you, pull out exact numbers from a mixed sample and really go into your downstream applications with high confidence. So not just for double-stranded DNA, we have dyes for RNA as well. The dye in this kit actually has a really nice selectivity for large or highly structured RNAs, right? So your ribosomal, your messenger, your tRNA, over either your small or disordered RNAs, right? So your single-strand RNAs, your maybe your siRNA, your microRNA, RNA homopolymers, or extremely degraded total RNA samples as well. This one is really nice, and it's kind of a unique one in its field. Most nucleic acid dyes that tend to bind to RNA will also have pretty high affinities for DNA as well. This one has really been a pleasure to work with. Of course, we have, as I mentioned before, right, the microRNA and single-strand DNA kits. These are nucleic acids that have a really, really high affinity for even the single-stranded nucleic acids, which is really nice. Again, it's this whole family of dyes has been super useful. People always have issues of trying to measure these in, I would say, single members of a mixed sample, especially for biomolecules, especially when people are working with really, really small amounts and have really important things that they want to do with these nucleic acid samples. And one of the things we were actually able to do with this entire platform of kits, the quantit and the quantitation kits, is they were so popular and people absolutely love them. So we built an entire small platform around them. These little flowers are called the qubit. So there's a qubit 4, as you can tell, it's the 4th iteration of the instrument. We also have one now that is the qubit flex, lets you measure 8 samples at one time. Basically, these are microvolume fluorescent fluorometers. They have 2 channels, 2 excitation channels, 2 emission channels, right? Your typical like blue excitation, green emission, then red excitation, far red emission. And we've actually got them nailed down to the point where you don't need a full standard curve. You can measure it with 2 standards, basically a bottom and a top. The curve is really, really reproducible, so you can get by with just those 2 standards. And it allows you to actually just really quickly measure it without having to go to a plate reader, without having to go to a fluorometer, prep a bunch of solutions. You can just add the reagent, add your sample, take a measurement, move on with your day and with what experiments you actually want to do. And the nice thing about having the qubit is we have full control over how people are interpreting the results, what the results are yielding. And so I'd be remiss if I didn't give a shout out to one of the first products I worked on whenever I came to the company. Is an idea I had, I think, as an intern. We started actually putting it into practice as a launched product. We call this the Rna Iq Assay. It basically uses the 2 different channels on the qubit, 2 different nucleic acid dyes in solution to give you a really quick check of an RNA sample, right? So there's a red, far red emitting dye that actually binds to your large intact structured RNA, a green one that binds to your broken down bits of RNA, your little bitty nucleic acid pieces. And this gives you a different range of emissions. And what we're able to do is kind of map those different emissions onto a quality score, where if your RNA is absolutely destroyed, it'll give you a really, really low quality score. It's kind of a quick check. It lets you give a really, really quick analysis of your RNA sample and say like, okay, is my RNA totally garbage, or is my RNA something I could probably take on down and maybe work with a little bit? So, that's kind of my spiel. In closing, we've been able to develop a really huge portfolio based on nucleic acid analysis. There's a lot more I would have loved to get into. I don't really have the time, but in all honesty, I kind of prefer the back and forth discussions more so than the presentation. The presentations are fun, but really I like to get other people's input, right? This is why we're scientists. And the whole reason we're scientists is to either publish papers or put out products there that other people use, make both of our science go farther and farther. So, we've been able to do a ton with these scaffolds. We've used them as switches for protease triggers, can put them on cells to measure viability, to measure various aspects of cell health, help people measure concentrations of things, improve gel electrophoresis. We're continuously looking for new applications, new avenues for these scaffolds. And the thing is, we always just want to talk to folks to see what they want to do with nucleic acid dyes, what they might want to do, how we could help each other to develop new dyes for those applications. So with that, let's talk. If anyone has anything they'd like to talk about.
Thanks, Chris. That was really interesting talk. And I think because of class schedules, this year's internship program students probably weren't able to join. So I'm wondering if I can put you on the spot to re-give that talk later this summer or early in the fall for those students when they start the immersion lab, because that was right on point for that program.
Oh, sure. Yeah, absolutely. I mean, it'd be good for them to see that stuff. Absolutely.
Absolutely. Yeah, it might start some discussions with them as well. With that, let's see if I look in the chat. Are there any questions for Chris?
I have a question. So one of the challenges we have when we're looking to prep our guts for flow cytometry or FACS analysis, the thing with epithelial cells, no matter the tissue, is they like to be next to each other and stick together. And when they're separated, there's this catastrophic rate of death. And so our flow experiments are always, with this super high death curve where we're throwing a bunch of PI in there and then trying to figure out, of those that are still alive, because they're not happy not being connected to their partners, what can they tell us? And so, but it's basically, it takes us a really long time to be able to, I would love to be at a point where we could be able to put some of these dyes in our preps and just have a check and see like if we're going to have a good flow day, or if I'm going to spend 5 hours on the sorter to get not a high yield because the prep was junk. And it's highly variable. It doesn't seem to matter who preps the cells. It could be me who has 20 years experience. It could be my grad students who have 5. And it seems to be the age of the gut. There's a lot of things that go into it, but I'd love to know if there was a product we could use, first of all, that would allow us to see, of course, just live dead beyond just the simple like blue colorimetric dyes we have right now. And then also, is there anything that can stay in there that allows us to see a shift to dead over time? Because if we have 4-hour sort, things are dying as they sit there. And so what kind of data am I getting in the first hour versus the 4th hour? So these are the things we think about because it's probably the most highly variable part of our science.
Yeah, absolutely. I totally understand. So a lot of our live dead kits would probably be really useful for that. I don't know if you want to do monocolor or dicolor. So there's ones that like the one I showed, right, that shows like green for all and then red for one or something along those lines.
Oh, okay, yeah.
I think some of those live dead kits would be super useful for you. Now the issue is, unless you want to have a lot of free dye in solution, which cytometry can sometimes handle that, and that's okay. It's usually like a one and done kind of deal. Like you can sometimes watch it over time if you have a lot of free dye in solution to still, because it's all based on the membrane permeance, right? And that's usually what's showing your compromised cells. Now, if you want something that will stay in the cells and indicates their viability over a long period of time, you would have to try it and see if you can maybe do it without a wash procedure or something along those lines. And so if you could have it in there in the solution that they're sitting in as they're going on to the cytometer, right?
So the other, I'll just ask a quick follow-up because I know there's other questions, but one of the initial assays for stem cells that was done before we had markers of any stem cells was the idea that they efflux dye. And so they end up being the, like this, it's called side population sorting, where you end up basically blocking the channels that can efflux and dye. And it turns out that those cells are still, I'm just wondering if there's any history of being able to, like, we are to the point where we understand what is marking certain stem cells, but we're really fuzzy in the transitions between a bona fide stem cell population and a non-proliferative epithelial population. And there's like a gradation in there of how frequently they turn over, and they might have different rates of turnover that might mean something physiologically, but we have no way to separate them. So I've been thinking about doing like differential side population sorting, sort of resurrecting this early 90s technique. But I was thinking when you were presenting about your dyes that there might be something that we could get there that would allow us to maybe see a differential, or is it always just like there or gone?
I mean, that's a really, really interesting question, but I might have to pass it over to people who are more on the biology side, in all honesty.
Sure, yeah.
Anyone else want to jump in on this one?
Hey, if you're looking for stem cells, do they have alk-phos activity? Because we definitely have a substrate that will use that.
Oh, that's a good point. Yeah, I mean, the thing is it's unknown whether these sort of further differentiated progenitors do. We know that the stem cells do. So so that would be one thing, but.
It's an option. Yeah, and then that'd probably be a pretty long conversation because we need to sit down and like kind of parse out, what are all the different physiological features? What could we leverage from the dye side? Not just nucleic acid dyes, but across any flora.
Across exactly. Yeah. Okay, thanks.
Yeah, absolutely.
Chris, I think maybe there's a lab watching party on the Eugene Thermo site, because there's an EUG lab group that posted a couple shout outs for you while you were going. And I know it's sometimes hard to see the chat while you have screen sharing on. So the first one was to shout out to the brand new Cytox Deep Red, which is they say is the best red nucleic acid stain we have.
Oh yeah.
And then also, as you were answering the question to Annie, suggested maybe a cell tracker dye or calcine green.
Those could be options, right? Yeah, and I guess that's getting back to overall valuability, right? So calcine green it's a fluorescein-based fluorophore that activates from esterase activity. So it's a classic live dead type assay. Esterase is pretty ubiquitous, right? Now cell trackers are really just a dye that goes in and labels a bunch of protein in your cell. And it's actually pretty cool because it will stick around for a long time. So you can watch it through several generations. I think through 4, maybe 3 generations. I can't remember the exact number that you can watch, but it'll stick around for a very long time. So you can look and see how many divisions they do. I'm not sure if a cell tracker would be super useful for differentiation, but it could be.
Maybe a Q tracker non-targeted that might leak out when the cells die. That's what I'm thinking.
Could be an option too.
That's a really good idea. Well, I've used cell tracker before, and the hard part is that sometimes I feel like it makes the cells behave funny, which is not what we're going for. But
yeah. And I see Vicky's question there. So Vicky, I tried. It's a little bit tough because a lot of the same structural features that I think this dye binds to are lost whenever you're running through a gel. So if it was a super non-denaturing gel where there was very little denaturing prep of your RNA, potentially, it's not something we've investigated a ton though.
Yeah, I'm just thinking, hi, Chris, there's like, sorry, I'm in a random place. I don't want to use the camera. There's just so many times when we really would like to know if, so we're working with some subdomains of the SARS-CoV-2 RNA.
Mhm.
And it'd be interesting, we'd like to know if it's folding or not folding. And a non-denaturing gel, you get bands, but you don't actually know if they correspond to folded or cleaved RNA. And then eventually we'd like to look at some folding patterns inside cells. And so usually people say, oh, we'll do like FRET or something like that, but that's kind of complicated. So it'd be really nice to have a like one size fits all structured RNA dye that worked under the conditions.
Oh, absolutely. Yeah, I've worked with this dye a ton. I have a good feeling for like when it turns on, when it doesn't, what structural features are important, though we haven't 100% nailed it down. If you want to work further on that, I'd love to send you some.
Score. That's great. We'll talk. That's wonderful. Thank you.
All right.
Any other questions for Chris? And Chris, I got a note about the watching party. It's just it's Skylar down in the lab. So I thought I recognized your voice,
yeah.
Skylar, but I wasn't 100% sure. So hi. Okay, so we will keep the channel open for a little while still. If folks want to hang out and chat informally, we can join Skylar in the lab perhaps and see what's going on at the bench. And keep in mind that we will be holding this symposium again in a month. So keep an eye out and email for upcoming talks in this series. David, any closing comments on your end?
No, just Chris, thanks also for sharing your work with the extended team, and it'd be great for you to connect with Vicky on the RNA pieces, and Annie, maybe there's some ideas that we might have for you as well on some other less dyes that could be metabolically used that aren't going to tell you live dead, but might give you more of a qualitative look at some of those preps also. So maybe we can catch up on that offline. Thanks, everyone.
Yeah, thanks everybody for joining us. And thanks, Tony, again for organizing.
Of course. Yeah.
Chris,
and I'm sorry.
Oh, no, go ahead.
I was just going to say, yeah, if we can stay on for a while longer, or else if there's no other kind of comments or questions, probably let everyone get back to their day.
Yeah, I saved one question for you, Chris. I just was curious that homodimer structure bound to DNA is super cool.
I know, right?
Is that, do you know binding affinity or any sequence specificity?
Yeah, we've that's known, and it was published. That's from Molecular Probes days, right? So whenever they had a lot more academic type partnerships, there's a few papers around that I could send your way. I'll have to dig them out. But yeah, they have measured KDs for those.
Okay, I was just making some connections. We just last week had our virtual symposium for the International Symposium on Macrocyclic and Supramolecular Chemistry, so ISMSC, and it'll be in Eugene next year. It was supposed to be in Eugene this year. And one of our speakers was Amanda Hargrove from Duke, who's doing some very cool small molecule binding to RNA. And so could be a good chance to interface with some of the Thermo folks next summer. There'll be a few folks around in that area, so we should be sure to do some connections. I'll get you the registration information as the time nears.
Absolutely, yeah, that'd be awesome.